Hi, I'm Allison Kokal. I'm a rising senior at the University of Illinois, and this summer I've been looking at mobile signaling. So mobile signaling is a process in which signals travel in response to external stimuli. So I'm specifically looking at guide RNAs. Not much is known about RNA transport, and so I created a dexamethasone inducible system that will create a visible phenotype within the tomato plant that will be able to see where these RNAs are traveling. And so I have two systems. I have my driver and control vector system and my driver and experimental vector system. In my control vector system, we expect to see the white sectoring only in the roots, but in the experimental vector system, we expect to see white sectoring in the roots and in the shoots because this construct has a mobilization tag that allows a CRISPR-Cas9 and guide RNA to travel from the roots to the shoots and make those edits. And so this summer, I have been crossing our T0 plants in the greenhouse to create our T1s. I cross our drivers with our control effector plants and our experimental effector plants, get plants that have our driver and control effector and our driver and experimental effector. And I did create a video on crossing that shows how I made my crosses this summer. That is on my YouTube if anyone wants to watch that. And then I also did genotyping of our T0 plants to make sure that those plants have our construct. And so I would take a tissue sample from our plant, I would extract its DNA, run PCR, and then I'd run a gel. And the absence or presence of a band would tell us whether or not the plant has our construct. And so from our genotyping, we're able to see that 11 out of our 16 driver plants, 12 out of the 13 control effector plants, and 16 out of the 19 experimental effector plants have their respective construct. And then here's an example of one of the gels I ran. On the right is our primer sets for our plasmid. This test for the plasmid, it is not specific for driver or effector, but we did test primers that will tell specifically whether or not the driver or effector is there that we'll use to genotype our T1 plants to make sure we have both of the constructs. And then we also run a primer set for actin. Actin is naturally produced in tomatoes, so we should see it every time. And it's just a control to help us with troubleshooting if the PCR is not working. And then from our crosses, we've gotten 16 fruits that I've collected the seeds from. And we germinated 4 of those seeds a few weeks ago. And then on Monday, we took a couple cuttings from 2 of those plants and we plated them on dexamethasone media. And after about 20 hours, we were able to see white sectoring in the leaf. And so this is building to our final system where we'll have our transgenic rootstocks with our CRISPR-Cas9 and guide RNA. And we'll graft that onto wild type shoots. And we'll be able to see white sectoring in the shoots because our guide RNA and CRISPR-Cas9 will travel from the roots to the shoots, making those edits. We'll be able to show that we can make transfer edits in the wild type shoots, and we'll be able to see where these RNAs are traveling.
So when right now you're looking to graft between 2 tomato plants, do you think the results would be similar if you were grafting between different species?
I think that would have to depend. There are 2 types of grafts. There are incompatible grafts and compatible grafts. So if you're grafting between 2 species that are incompatible, probably wouldn't see the same effect. With 2 compatible species, possibly it'd have to have like similar genome that has this gene that we are cutting out that will make that visible phenotype.
Thank you so much.